Good afternoon, children. In the last class, we have completed the very important topics, that is a transcription and translation. Today we will continue with respect to the further important topic, so that is the regulation of gene expression. It is also known as LAC OPERON concept. Okay. See, when it comes to the LAC OPERON concept, first of all, just recall. Okay, so how the process it was begin from the transcription towards the translation. You know very well that the proteins are the building blocks of an organism synthesized in a cell as they are directed by DNA, isn't it? So that means the proteins are building blocks of an organism synthesized in a cell as they are directed by DNA. And the protein molecules are produced when the cell requires, and they are not produced when there is no necessity of the proteins. So that means whenever the requirement is there, then only protein synthesis will be takes place, or else there is no necessity of the protein, so that there will be no the synthesis of protein also. Okay, so are you getting what I'm telling? The protein molecules are produced when the cell require, okay, and they are not produced when there is no necessity of the proteins. Okay, so hence the DNA undergoes a switch on and switch off mechanism for protein synthesis. So this is also called differential gene action. So this process is explained by, so that is Jacob and Monod. So that's what we call N generally in the form of regulation of gene expression. Okay, so let us discuss about the topic, the regulation of the gene expression. See, this regulation of the gene expression, especially in eukaryotes, occur by the following levels. So here only indication. Okay, so observe the properly. In eukaryotes, the gene expression occurs by the following levels. Okay, so it is majorly we can identify in the 4 levels. That is transcription level. It is also known as the formation of primary transcript. The processing level, it is also known as splicing and capping. So that's what we were discussed earlier. Okay, I hope you can remember. Then the transport of messenger RNA from nucleus to the cytoplasm. And finally, the translation level, the formation of a polypeptide. So these are all things we were discussed already, isn't it? So what are those? The following levels: transcription level, processing level, messenger RNA transport, and finally translation levels. Okay, here only if you if you can identify, so these are all where exactly happens, that was indicated. Okay, so that means inside the nucleus, gene on DNA and heteronuclear RNA, messenger RNA, these are all the synthesis will be takes place. Outside the nucleus, that means inside the cell, but exactly in the cytoplasm. Okay, so in the cytoplasm, outside the nucleus, the protein synthesis will be takes place. Okay, so let us discuss how this process exactly takes place in the eukaryotes. Okay, you know, the metabolic that is a physiological and environmental conditions regulate the gene expression. Okay, so how the gene regulation will be takes place? So that means the regulation of the gene expression, it is directly influences the metabolic, physiological and environmental conditions which regulate the gene expression. Example, in Escherichia coli, the beta galactosidase enzyme hydrolyzes lactose into galactose and glucose. Okay, so in the absence of lactose, the synthesis of beta galactosidase stops. Okay, so it is also known as a switch off mechanism. I will tell you then. The development and differentiation of the embryo into the adult are the result of expression of several set of genes. Okay, So that means the development and differentiation of the embryo. If we take example of human embryo only, in adult are the result of the expression of several set of genes, not only single or only two or a few genes. It is because of the, that is a several set of the genes. They are responsible for the formation of the different of the, the organ systems which is present in the body. Okay. And here if you see that the process, if a substrate is added to growth medium of the bacteria, a set of genes is switched on to the metabolizers. It is called induction. Okay, it is called induction. Remember, it is also known as the inducer. Are you getting? I'm telling the basics things to understand the LAC OPERON concepts. Okay, please concentrate properly. If a substrate is added to the growth medium of the bacteria, a set of genes is switched on to the metabolize. It is called induction. It is also known as the inducer. Okay. When a metabolite, the product is added, genes to produce it are turned off. It is called repression. Okay, repression or repressor gene. Okay, remember all these things. Okay, so these are all the basics to understand the regulation of the gene expressions. Are you getting? When a metabolite or the product is added, the genes to produce it are turned off. This is called the repression or the repressor genes. Okay. See here only, if you see that, first of all, as I said, the regulation of gene expression was explained by the LAC OPERON concept, or the LAC OPERON concept, which helps to understand the regulation of gene expression clearly. Then the question arises, what is the operon? Okay, so each metabolic reaction is controlled by a set of genes. Okay, so whatever the metabolic process, each metabolic reaction is controlled by a set of genes. All the genes regulating a metabolic reaction constitute an operon. We are getting a cluster of structural genes together with operator gene is called operon genes. Example, okay, if we take a example, LAC operon. Okay, there are different type of operons comes. Tryptophan operon, ARA operon. Okay, so then his operon, valin operon, like that there are different type of the operons comes. But in our syllabus, they have given only the one concept, that is only LAC OPERON concept. Okay, which is try to explain how exactly the regulation of the gene expression takes place. Okay, so let us discuss about the LAC OPERON concept. First of all, okay, so how this process actually, okay, so it was explained by, that means the LAC OPERON concept was explained by, there are two names of the scientist, that is a Francisco Jacob and that is a Jacqueline Monod. Okay, so it was explained by Jacob and Monod in 1961. Under LAC OPERON concept that explains the regulation of the gene, that means how the regulation of gene action takes place. They conducted the experiments on the utilization of the lactose by E. coli bacteria. Okay, so that means the LAC operon is the operon controlling lactose metabolism. Okay, so that's why they named their concept in the name, that is a LAC OPERON concept. Are you getting? Fine. And here you can identify when the lactose molecules are absent in the nutrient medium, the DNA of the bacteria is switched off and do not produce any enzyme proteins. And when the lactose molecules are present, the DNA of bacteria is switched on to produce enzymes which are necessary to break down lactose into glucose and galactose. Okay, yeah, the DNA of the bacteria consists of several genes arranged in a particular manner. Here only I hope you can identify they are a regulator gene. I is a regulatory, that is a regulatory gene, and the promoter gene P, capital P, and the operator gene O, and the structural genes Z, Y, A, they are called structural genes. Are you able to identify? Okay, and I told you earlier also the what is the meaning of operon? The cluster of structural genes together with operator gene is called operon genes. Okay, here only you can identify the LAC operon consists of a regulatory or inhibitory, that is inhibitor I gene, which codes for repressor protein. Okay, and the three structural genes, small z, y, a, that is a z gene, y gene, and a gene, and that is a operator region. Are you getting? I hope you can identify easily. And the genes in the operon functions together in the same or related metabolic pathway. So then only the successful regulation of gene expression with respect to the particular protein synthesis will be takes place. Okay, yeah. So here only, so they have given first of all how they play the role. Okay, see, the enzyme RNA polymerase always binds to the promoter region by identifying the specific nucleotide sequences, that is in generally known as TAT. So this region is also called Tata box or Pribno box. Okay, and when the RNA polymerase enzyme transcribes the structural genes that produce the three messenger RNA strands, these the messenger RNA strands undergo translation to produce the three enzymes such as Z gene produce galactosidase, that is a beta galactosidase, Y gene produce beta galactosidase permease, and A gene produce beta galactosidase transacetylase, and these three genes together break lactose into glucose and galactose. Are you getting what I'm telling? See, this can also be asked in the form of 1 marks or 2 marks. Write the role of structural genes, like that also they can ask the questions. So that means the Z gene codes for the beta galactosidase enzyme. It hydrolyzes lactose to galactose and glucose. Y gene codes for permease. It increases permeability of the cell to beta galactosidases, that is lactose. And A gene codes for transacetylase. Okay, so this is how exactly, so the structural genes, they play a very vital role during the regulation of the gene expression as well as the synthesis of the protein. Okay, and see here I told now, so whenever, so that means how can we classify as a switch on and switch off mechanism? Okay, it is based on the DNA undergoes switch on and switch off mechanism for protein synthesis. It is called differential gene action. But how can we say a switch off mechanism, or how can we say switch off or switch on? Okay, let us discuss one by one. In this one, the first one is, if you in the absence of end user. In the absence of end user, it is also known as switch off mechanism. How it will be? Okay, see, observe the properly whatever they have given the the genes. Okay, so it is containing promoter, regulator gene, and operator gene, the structural genes, so and repressor, messenger RNA, and repressor protein. Okay, so when will we say the switch-off mechanism. When lactose molecules are absent in the nutrient medium. Okay, so whenever the lactose molecules are absent in the nutrient medium, DNA is switched off for protein synthesis. Or we can also say, if there is no lactose or the end user, the LAC operon remains switched off. Okay, and the regulator gene produce a kind of protein is called a repressor. Okay, so what is a repressor? The regulator gene produces a kind of protein called a repressor. So that binds to the operator gene. Okay, immediately, that means the repressor binds to the operator gene. These do not allow RNA polymerase enzyme to transcribe the structural genes. Okay, remember, whenever the lactose molecules are absent in the nutrient medium, the DNA immediately, DNA is switched off for the protein synthesis. The regulator genes produce a kind of protein called repressor that binds to the operator gene. So this do not allow RNA polymerase enzyme to transcribe the structural genes. Hence, there is no transcription. The translation and required the protein is not synthesized. Okay, so hence it is called switch-off mechanism. Okay, hence it is also called switch-off mechanism. So which can also be known as in the absence of end user. Okay, are you getting? Yes. And what about the another case? As I said, in the presence of end user. Okay, as I said, when we can see the presence of, the presence of end user, it is also known as switch-on mechanism. When the lactose molecules are present and the nutrient medium, DNA is switched on for the protein synthesis. Are you getting what I am telling? Okay, here the lactose play very important role. When the lactose molecules are present in the nutrient medium, the DNA is switched on for protein synthesis. The lactose molecules are in the nutrient medium, acts as an end user. Okay, so acts as an end user which binds to the that is a repressor to form end user repressor complex. Okay, so this is very important. So you should identify here. So how the end user repressor complex will be formed? See, the lactose molecules are added in the nutrient medium. Whenever we add the lactose molecules into the nutrient medium, which is acts as an end user and which binds to the repressor to form an end user repressor complex. Are you getting? Now, the repressor become inactive and release from the operator gene. So this enables the RNA polymerase enzymes to transcribe all the three structural genes, that is ZYA. Okay, are you getting what I'm telling exactly? Okay, fine. And the process will continue. How it will start? So as I said, whenever the operator regions becomes free and the end users, the RNA polymerases binds with the promoter gene, then the transcription starts. Okay, fine. And the process will continue. Okay, so how the process will continue? So the repressor becomes inactive and releases from the operator gene. So then enables the RNA polymerase enzymes to transcribe all the three structural genes, that is ZYA. Are you getting now? So this results in the synthesis of three messenger RNA molecule continuously, or called polycistronic messenger RNA. Okay, they are also known as polycistronic messenger RNA. Fine. Now all these three messenger RNA strands undergo translation to produce three polypeptide chains or three proteins which acts as a three enzymes such as beta galactosidase, permease and transacetylase. Okay, so these three enzymes together break lactose into glucose and galactose. Are you getting? So that means the glucose or galactose cannot act as an inducer for LAC operon. The regulation of LAC operon by repressor is called negative regulation. Negative regulation. Now the LAC operon is under control of positive regulation as well. Okay, so that these three enzymes together break lactose into glucose and galactose. Fine. Now the polycystonic messenger RNA, the product of the first gene activates the second gene, and the product of the second gene activates the third gene, and so on. So this is called pre-programmed circuits of the gene action. Okay, are you getting? So this is how all the genes are interrelated, interconnected, interdependent on each other. So that means if the first gene activates, then only second gene activates, and then later third one. If there will be no activation the first gene only, there will be no activation of the further genes. So that's what I said. In polycystonic messenger RNA, the product of the first gene activates the second gene, and the product of the second gene activates the third gene, and so on. So this is called the pre-programmed circuits of the gene action. Okay, so all these things, so this was explained by the name of the scientist Jacob and Monod. So and they took the example, okay, under the LAC operon concept. They tried to explain the regulation of the gene successfully, and whatever they conducted the experiments by utilizing the lactose by E. coli bacteria, finally they given the complete explanation about the regulation of the gene expression by using LAC operon concept. Okay, so this is what till now one of the successful operon concept whatever we are studying now. Okay, so and also it is so this LAC operon concept successfully explains how exactly the protein synthesis takes place in the eukaryotes also. Okay, are you getting? See, please remember, so this is very important. You have to write and practice the switch off and switch on mechanism. Surely in the examination they will ask the question in the name of, so give the LAC operon concept, that means this is only, or sometimes they will ask give the regulation of the gene expression, or sometimes they will ask the specifically write the switch on mechanism or the switch off mechanism, like that. Okay, so remember, so this is very important. Okay, it's very clear, and actually if you understand properly, this is very easy. Okay, fine. So when the lactose molecules are absent in the nutrient medium, the DNA of the bacteria is switched off and do not produce any enzyme. Okay, so that means the proteins. When the lactose molecules are present, the DNA of the bacteria is switched on to produce the enzyme which are necessary to break down the lactose into glucose and galactose. Okay, so almost all the diagrammatic representation, the whatever the regulator gene, operator gene, these all will be same. But while you are writing in the absence of inducer, in the presence of inducer, you should know. Okay, so after the the repressor proteins becomes active and inactive, and the repressor binds to operator region and prevents the RNA polymerase from transcribing the operon, which is comes under in absence of inducer. That means the switch off mechanism. In presence of inducer, so that means the repressor. Okay, repressor, that means inducer, it becomes a inactive repressor, and there are three names of the enzymes you should remember. So that is beta galactosidase and beta galactosidase permease and beta galactosidase transacetylase. Okay, so these three, these 3 genes you should remember with respect to the process. Okay, so then only you will get the full marks also. Based on this, they will also ask the questions in competitive also. Okay, so first of all, try to write down this one, the structure, and also you should write and practice how exactly the switch off and switch on mechanism will be takes place. Okay, so this is about the LAC OPERON concept, or it is also popularly known as regulation of the gene expression. Okay, so let it be. Next class we will continue further topics. Thank you. Have a nice day.